Okay. All right. Mark is one of my colleagues at Memorial Sloan Kettering. He's a pathologist by training, but his lab has been extremely busy developing genomics-based therapies, identifying mutations in tumors that would potentially be targets, looking at, in particular, sarcomas, non-small cell lung cancer, such as EGFR mutations, and most recently, some exciting data about BAP1, a gene in mesothelioma, and so he's here today to talk about that.
Well, thank you, Lee. It's a pleasure to be here. I was also very glad to see how generous and delicious the breakfast break was, because I know I'm standing between you and lunch, so this is always a bad situation, but hopefully you enjoyed the nice food at the break. So, so we're going to change gears a little bit. I guess really I want to tell you more about some more basic work that we do to understand mesothelioma, what goes wrong in mesothelioma cells, and more recently, what may predispose some individuals to mesothelioma. So just to kind of set the stage, I think it's good to, make a very clear distinction between two types of cancer genes, and this gets confusing very often. So they're genes that are mutated only in the cancer cells, not in the normal cells of the patient. And those we call typically somatic mutations or acquired mutations. And for instance, in mesothelioma, those might occur accidentally, or they might be due to specific carcinogens such as asbestos. And typically, if that mutation confers a growth advantage to the cancer cell, to the cell, that cell may eventually evolve into a cancer. The second type of cancer gene is a gene that is mutated in the normal cells in every cell of your body that somehow predisposes you to particular types of cancer. So good to keep these two, make a very clear distinction between these two types of cancer genes. But as you'll see, sometimes one type becomes another type. So we'll start with the somatic genetic abnormalities in cancer. So there's been a great effort at trying to understand what exactly are the genetic alterations in cancer cells, and it's been shown that if you can understand those better, in many cases you can actually devise specific therapies to treat those cancers. And each cancer type has a different spectrum, a different range of genetic alterations. Some of the common types of genetic alterations in cancer are shown in a very schematic way here. So copy number changes, meaning your cancer cells have an extra piece of some chromosomes, or they lose some pieces of other chromosomes. Genetic rearrangements, mutations, so changes in the sequence of your DNA in specific genes, changes in the expression of specific genes, as well as other changes that regulate gene expression. And the research interest and the clinical interest in doing this, of course, is the example set in other cancers. In the GI gastrointestinal cancer called gastrointestinal stromal tumor, there's specific mutations in KIT, and there are dramatic responses to a KIT inhibitor, imatinib. In lung cancer in 2004, it was discovered that lung cancers that have mutation in the EGFR gene are very responsive to inhibitors of the EGFR gene. And most recently in melanomas, about 50% of melanomas have mutations in this gene called BRAF, and they are very responsive to the specific inhibitor of this mutant BRAF. And these scans show the before and after pictures for these patients. So this is the model that is driving a lot of research in the field. So we have to match patients to drugs based on the mutations that drive those particular tumors. And it's really kind of has driven a revolution in oncology and in cancer research. You can see some of these news articles in the scientific literature. So turning to mesothelioma, so we've been working on mesothelioma for many years, actually, and working on it from the point of view of working on human mesothelioma tumor samples and cell lines. So obviously, the fact that patients agreed to donate some of their tumor for research is extremely important to this kind of research. And so looking at the genetic abnormalities in mesothelioma, I just want to give you kind of a view of, you know, how messed up the chromosomes can be. So this is your typical male karyotype, you know, 46 chromosomes, XY, everything is, well, this has been, of course, arranged by somebody who studied the chromosomes. But in a cancer cell, the chromosomes are very abnormal, multiple copies of some, complete loss of some other chromosomes, and there's some chromosomes that are so messed up that you can't actually tell which chromosome number they are. So we used methods to study these genetic abnormalities in tumors, in mesothelioma tumors. And we did this using 53 mesothelioma samples from our institution, as well as mesothelioma cell lines, and this was a collaboration with Bruce Johnson in Australia. And we looked at the changes in DNA content in these mesothelioma samples in order to try to understand better the biology. And this is about the sum total that was known about the genetic abnormalities in mesothelioma at that point. It was well known that almost all mesotheliomas lose this gene called P16, and many of them lose this gene called NF2. So, and I'm really compressing this story, but what we did was we took these 53 mesotheliomas and we measured the DNA content for each chromosome in these 53 samples, and then tried to understand if there are some parts that were more often gained than by chance and some parts that were more often lost than by chance. And this is shown here. So here you have your chromosomes lined up from chromosome 1 to 22, kind of end to end. And the parts that were significantly gained are shown on top, and the parts that were significantly lost are shown on the bottom. And this is the kind of the sum total of 53 tumors. And the idea is that the parts that are often gained may contain what are called oncogenes, and these genes may sometimes be also activated by point mutations in their sequence. Conversely, the parts that are often lost in tumors can contain tumor suppressor genes, and these can be inactivated by other types of mutations in their sequence. So the point of this one then was to look at these regions, look at the genes in these regions, and pick some that might be interesting for sequencing. So we did, since this was the most dramatic loss is the P16 gene, which was previously known. We didn't sequence this. This was already well known. But we did pick 25 other genes for sequencing based on their location in these regions and possibility that they're the kinds of genes that could be mutated, including the NF2 gene, which is on chromosome 22, as you can see, is frequently lost, or at least one copy is lost, and the other copy may be mutated. So the most interesting gene that came out of this turned out to be a gene on chromosome 3. So that's over here, chromosome 3. In this region, that's often lost, a gene called BAP1. And when we looked more closely at the region of this gene, so in this display, each row is a different patient's tumor, and it lines up the regions that were lost in these different patient tumors just in this part of chromosome 3, and it really lines up on the BAP1 gene. And when we looked at the DNA sequence of the gene, about 20 to 25% of cases had mutations. Another 30% had actually loss of a copy. So we're estimating that about 40% of cases have some inactivation of this tumor suppressor gene, making it the third most frequently altered gene in mesothelioma after P16 and NF2. And this is another way of displaying this information. So here are the genes that had mutations in the study. And in this instance, each column is a tumor from a single patient, and it shows you the mutations that tumor contained. So you can see that many patients had BAP1 mutations in their tumors, many had NF2 mutations and loss, and many had CDKN2A or P16 losses. So those being the three most commonly mutated genes in mesothelioma. Now, what does the BAP1 protein do? Actually, I'm not going to talk about that at all. This is one of the messages I want to leave you with is that this is so new that there are still many more questions and answers. So BAP1 seems to be involved in different cellular processes. Now, at this point, we don't know which is the key process that makes the loss of this gene significant to the biology of mesothelioma. So obviously work is being many groups are pursuing work on understanding what BAP1 does in the cell. So we published these results in June of 2011, and then things got really kind of interesting. While we were working on our story and submitting it for publication, another group led by Bill Harbour identified mutations in the same gene in ocular melanomas. Ocular melanomas, you think mesothelioma is rare. Ocular melanomas are about half as frequent as mesothelioma. So it's an unusual tumor. And the mutations were very similar types of mutations to the ones that we saw in mesothelioma. Obviously, we could not. We couldn't think of anything that these two tumors have in common, other than these BAP1 mutations. Now, just getting back to the distinction between germline and somatic mutations. So again, so far I've been telling you about mutations in BAP1 that occur in the cancer cells only, not in the normal cells of those patients. But where the story really got interesting was that it turns out that BAP1 also can be mutated rarely in the normal cells of patients as a germline mutation. So the group led by Joe Testa and Michele Carbone identified two families with mesothelioma in multiple generations, and these families carried a mutation in the BAP1 gene. At the very same time, a group from, partly from our center, identified families with multiple generations of ocular melanoma that also had mutations, germline mutations in BAP1. And since then, there have been several other reports. So here's an example of the families from Joe Testa's report. So here you can see, I've indicated, just to simplify, with the red arrows, the patients who, the family members who had mesothelioma. So you can see this very intense familial clustering of mesothelioma. Also note that some family members also had uveal melanoma, so the two do happen in the same families. Here is, I'm sorry, yeah, so here is another pedigree from one of the other reports, which was more centered on the skin abnormalities, but again, so this is the pedigree, this is the pedigree from another report, again showing mesothelioma in multiple generations. These patients, in addition, have unusual moles. So this has become a very interesting new information for dermatologists who screen patients for melanoma. So they have a distinctive type of mole that doesn't generally turn into a melanoma, but can be helpful in identifying these patients. So I just want to try to answer some questions that immediately come to mind now that we have this information about this germline BAP1 syndrome. So the first question is, what percentage of mesothelioma patients without a family history of mesothelioma have a germline BAP1 mutation? So if a patient presents with mesothelioma, what's the likelihood that the germline BAP1 mutation is responsible for that mesothelioma? We think it's very few. So the information from the initial study suggests that it's probably less than 5%, probably less than 2% of all mesothelioma patients have this germline BAP1 mutation. But again, this is a very new area and more studies are needed. We have a trial opening up at our center led by Marjorie Zauderer and Lee Krug to screen patients for germline BAP1 mutations to get this information in a systematic fashion. I'm sure this is going on at other centers as well. A second question, if you have a germline BAP1 mutation, what's your lifetime risk of mesothelioma? From the pedigrees, there were members of those families that had the mutation in their normal cells, did not develop mesothelioma. It looks like it's got to be less than 50%, but again, more families need to be studied. In all the papers that have been reported to date, there may be there's still less than 10 families reported, so we have very little information. We're basing our discussion on very little information. If you have a germline BAP1 mutation, can you develop mesothelioma without being exposed to asbestos? So this is unknown. We really need more studies. I think that the two families that were studied by the Joe Testa group had no apparent asbestos exposure. Again, this is hard to pin down, so really we need more studies in that regard. Do all families with germline BAP1 mutations develop both ocular melanoma and mesothelioma? They don't. So there are some families with more ocular melanoma and some families with more mesothelioma. You might wonder whether there's differences in exposure to asbestos versus ultraviolet light. Ultraviolet light, of course, being the risk factor for melanoma development. If you're healthy, should you get tested for germline BAP1 mutations? No, not unless you have a family member with ocular melanoma, very rare occurrence, obviously. Or this is just my kind of out of the hat suggestion is that if you have two family members with mesothelioma, that may be a reasonable thing to consider. But right now, if you just have one family member with mesothelioma, I think the first thing to do is to get that family member tested, and you don't need to get tested. But more studies are needed. So you can see more studies are needed. So just to go back now to the somatic mutations, the story keeps getting more complicated and interesting. I told you about this, the finding of these mutations in ocular melanomas, and they tend to be in ocular melanomas that are very aggressive and kill the patients. More recently, it's been identified in about 10% of kidney cancers and has also been associated with a more aggressive form of kidney cancer. And finally, just very recently, there was a report that linked a loss of BAP1 to myeloid leukemia, but only in mice so far. The BAP1 mutations appear to be extremely rare in human myeloid leukemias. The interesting thing here in these experiments is that this group inactivated BAP1 in adult mice, and the only tumor that the mice developed were myeloid leukemias. They did not develop ocular melanomas or mesotheliomas. Again, maybe suggesting that you need another carcinogen to trigger those additional cancers. But again, mice are not humans, and you have to take everything with a grain of salt. I just want to take a moment to tell you about some work that we're pursuing that's supported by MARF. And so one of the approaches that we're taking in our work going forward is to look at BAP1 mutated mesothelioma cells and understand to try to find out if if these cells acquire a vulnerability that's not present in normal cells. And this is based on this principle of synthetic lethality, which basically means that you might have a loss of a tumor suppressor that can enhance the growth of your tumor cells, but at the same time, it may make those tumor cells more dependent on the activity of another gene that picks up the slack in some cellular function. So this is shown schematically here, where you might have two genes that are necessary for cell survival. They fulfill some kind of function. If you block one of those genes, the other gene can still maintain that function. In cancer cells, what seems to happen then is you have a mutation in one of the genes. For other reasons, perhaps it leads to increased cell growth, but you're left with only the first gene that can maintain the normal cellular function, such that if you find that gene, you can trigger cell death because your tumor suppressor gene is already mutated. So this is something that has been explored in other cancers, in particular breast cancer, where there's some very interesting results. So what we're doing is doing a systematic screen of mesotheliomas with BAP1 mutations to find out if there are other genes that could be blocked, and by blocking those genes we could kill the BAP1 mutated mesothelioma cells. So there's obviously a large group of investigators and collaborators that work on this in our center, and listed the clinical collaborators as well, Lee Krug, Marjorie Zauderer, Valerie Rouche, and so on. And we're supported, as I mentioned, by MARF and by MSKCC Philanthropy. So just last little bit, I want to tell you about something else that's bigger than this, so to speak. So as I mentioned, we in our studies in our lab, we looked at 25 genes, and we found a very interesting new gene. One of these 25 genes was a very interesting new gene in mesothelioma. But it's possible that some of the other genes that are not frequently altered in their copy numbers might also be significant mesothelioma. We don't know. But I think now we have the opportunity to sequence every gene in every mesothelioma tumor to understand if there are any other genes that contribute to mesothelioma development. And here's a little timeline kind of what we've been able to do in the lab over the years. And so in the 90s, it was a big deal if you could, determine the sequence of one gene in a few samples or even a few dozen samples. That was a big deal. Then we went to technologically be able to develop, to establish the mutations or in many genes, in many samples. And now we're really able, because of technological advances, to take dozens and hundreds of samples, cancer samples, and determine the genetic alterations that are present in every single gene in those samples. And this is because of tremendous technological advances in DNA sequencing. So this is just to show you the cost per megabase. So the human genome is 3 megabases of sequence. So the cost per megabase in 2001 was $10,000 and is now about 10 cents. So there's been an amazing technological progress here that really makes it possible to do things that you couldn't even think of before. So there are these very powerful new sequencers called next generation sequencers that can basically sequence a human genome in several days to hours. And again, gives you an idea of how the costs have come down. So the Human Genome Project that was mostly done in the 90s cost $3 billion. Today you could do that for about $10,000. And the technology just keeps progressing. So these new sequencers that can sequence a human genome in a day. And the one that's kind of the coolest, of course, is a sequencer that you can plug into your USB port and sequence the human genome in a day. So obviously this is completely taken off. So the problem now is not sequencing, it's analyzing the data that are generated. So with that little kind of intro to this last part, I just want to tell you about this project that's getting underway that harnesses these new sequencing technologies, and that's the Tcga and Icgc joint project on pleural mesothelioma. So the Tcga is the Cancer Genome Atlas, which is a joint project of the National Cancer Institute and the National Human Genome Research Institute. I'll tell you more about it in a minute. The Icgc is the International Cancer Genome Consortium. So this is a coordinated global effort to get comprehensive genomic information on as many different cancers as possible. Obviously, I'm not going to go through all this, and many of the cancers are relevant to the countries, to specific countries. So kind of humorous, well, not humorous, but interesting that, so for liver cancer, China is focusing on, Japan is focusing on HBV-associated liver cancer, and they've teamed up with France, who's studying alcohol-associated liver cancer. So there's complementary efforts. The Tcga itself has selected a number of different major cancers for this project, and they're listed here. And the 3 original ones are in red and have already been completed, essentially. And these cancers required 500 samples of each to be available, very high-quality samples with matched normal tissue from these patients. More recently, the Tcga decided to establish a rare tumor project, and on the urging of several of us, mesothelioma was included in this project. So, and the original goal was 50, so the idea of this rare tumor project was that they would do 10 tumors, 50 samples each to make 500, but now they want to ramp this up to 200 tumors. And this is very timely because at the same time, there's a global effort that was trying to get underway, led by Bruce Johnson from Australia, Bruce Robinson from Australia, and so there's two efforts have now joined forces, the global effort to do this in mesothelioma. Just very briefly, these projects all have this kind of same pipeline where there's a very complete genetic characterization of every single tumor. This is the state-of-the-art for each tumor, far beyond what any individual lab can do, with an extremely sophisticated analysis of the results. So these have already, this has been cranking along. So this was the study of brain tumors that was published in 2008, the study of ovarian cancer published in 2011, and most recently the study of squamous lung cancers published a few weeks ago. So the criteria for getting samples into the study are very stringent. This has been one of the issues for mesothelioma. So they have to be frozen tumors, untreated, fairly large pieces of tumor, obviously a thorough pathology review, a high tumor content, no prior treatment. So this is a problem for many centers that use multimodality therapy. Matched blood from the same patient. Of course, all the approvals. So this is why to get to 200 tumors, we need to enter potentially 500 tumors into this pipeline to end up with 200 good tumors down here. So this effort is underway. And these are some of the contact points. The Tcga director at the NCI, who's very committed to this project, is Kenna Shaw. And the other Tcga representatives are Harvey Pass and myself. And then the international representatives are Bruce Robinson from Australia and Peter Campbell from the Sanger Center in the UK, where the sequencing for this project will be done. So I think this is a very exciting development, and I think I will leave you with that and take questions.